the Ethics and Global Health seminar series for 2009-2010. Professor Schneewind will be our concluding speaker in the series, and I'm going to tell you a little bit about Olaf Schneewind, who is the Louis Bloch Professor in Microbiology, Chair of the University's Department of Microbiology, whose long-term work has examined mechanisms and strategies of pathogenic bacteria causing human disease. In 2003, Professor Schneewind became the principal investigator of the Great Lakes Regional Center of Excellence for Biodefense and Emerging Infectious Disease Research. This was a heavily competed for program that supported a consortium of 20 institutions in the greater Chicago area, funded by the National Institute of Allergy and Infectious Diseases, and under Dr. Schneewind's leadership of his Great Lakes Regional Center has focused on developing vaccines and treatments and diagnostics for diseases such as anthrax, botulism, tularemia, hemorrhagic fever virus, and plague. Also, beginning in 2003, Professor Schneewind applied and was awarded a state-of-the-art biosafety level 3 facility, the Howard Ricketts Laboratory, that will be constructed at Argonne National Laboratory. That received full approval in December of 2009 and will be commencing operations. It's a great pleasure to introduce to you Professor Olaf Schneewind, who will speak today on vaccines for the developed and the developing world.
Thanks. Thank you, Mark. So I had to make a selection as to what vaccines and diseases I was going to talk about. Rather than talking about all diseases and in particular agents that concern with biodefense, I elected to focus on an emerging pathogen, that's Staphylococcus aureus and MRSA infections. Okay, so the talk is to some degree fairly technical, and obviously I spent a lot of time on these technical attributes, and unfortunately the technical attributes are essential to understand whether one can develop a vaccine or not. That is not to say that I wouldn't want you to interrupt me if you don't understand anything, or if there is any particular topic in the presentation that you want to discuss further. So I know the end of the talk, we have an hour and a half, I understand, at your leisure. So first I want to introduce the organism and the disease that I want to talk about, and it's a disease that affects both the developed as well as the developing world. And that organism is Staphylococcus aureus. It's a gram-positive pathogen that on transmission electron microscopy pictures reveals itself to us as a simple organism with a cytoplasm, a cytoplasmic membrane, and then a very thick cell wall. The organism is very small. It's about 800 to 900 nanometers in diameter, so it's significantly smaller than actually E. coli, for example. The organism is a commensal of people, and about 50% of the people in this room are colonized with the organism. And most of you will not have an infectious disease from it, say this year. What's interesting about the colonization of people with this organism is that in particular settings, but now also in the community, the organism has acquired resistance to antibiotics. And obviously then you will carry these organisms in your nose, in your nares, in your ears, under your fingernails, and on your skin. And any social contact that you engage in will lead to the transmission of the organism to recipients. In the hospital setting, those recipients can be our patients. And obviously that's how they then become infected. Typically, people are infected with the strains that they are colonized with. In the last 20 years, there's been a change in the incidence of staphylococcal disease, and this organism is now the leading cause of bloodstream, skin, and soft tissue, as well as respiratory infections in the United States, in the developed world, and now also increasingly so in the developing world. And I have a few comments to make on that. Now, hospital-acquired MRSA, but also community-acquired, that is CA, MRSA infections, occur with strains that share 80% of their genome, which is to say, the organism, in contrast to many other pathogens, is actually not highly variable. And there's a scaffold of the genome that is conserved between all of these strains. And that attribute in of itself actually revealed some of the interesting strategies that the organism has in causing disease. Now, the U.S. mortality for Staph aureus and MRSA infections for the last 3 years exceeds that of HIV, AIDS-infected individuals, or of any other infectious disease. And the trajectory for those diseases is increasing in time. So much so that we're now looking, and I think this is the first bullet point, on balance in American hospitals as a 4% infection rate for any hospital admission. It is, any person that comes into a hospital for whatever reason they may have, in 4% or perhaps soon 5% will acquire an MRSA infection. In hospitals across the country, that frequency of MRSA, where there's methicillin-sensitive strains, is somewhere between 70 to 80%. And in some hospitals it's worse, and in other it's less worse. This hospital is no exception from that rule.
Are you saying the mortality directly from the bug, or are you saying people die with the bug?
No, they die from typically a bloodstream infection. The life expectancy from somebody with a diagnosis of MRSA bloodstream infection is 50%. Now, an FDA licensed vaccine is not available for that disease. It's hard to put a number on the total morbidity in the United States. It's probably somewhere at 1.5 million in the United States alone. Worldwide, it'll be hundreds of millions of people that suffer from Staph aureus infections. Okay, now how does it look in the developing world? So I was recently at the Congress for International Infectious Diseases. Where was it again? Somewhere in Florida, I think. Anyway, and the key factor for MRSA infection and transmission in the developing world is the overcrowding of hospitals. So these two images, they capture very quickly that much of the social services that are provided in the hospitals stems from relatives. They crowd these hospitals day in and day out. The hospital beds are very close together, and an MRSA infection or colonization with one patient will then spill over to the other patient. So much so that MRSA infection rates in the developing world haven't been calculated, but estimated to be significantly larger than the 4% of the developing world, of the developed world. So while these people are admitted for various causes, surgical issues or whatever the reason may be, they once again in the developing world will acquire a Staphylococcus aureus infection, and that could then be the deciding factor in the outcome of their morbidity. Now, the key question is, what is the incidence of MRSA in the developing world? So the data for the last couple of years indicate that MRSA infection in Africa has now exceeded 15%. It's about greater than 25% in Southeast Asia, and that number seems to be similar to the numbers for South America. In South America, the first reports for community-acquired MRSA infections have been reported. So it appears that what was once a distinction between the developed world and the developing world, namely that the developed world had MRSA infections and the developing world did not, that distinction is disappearing to a degree that there will be no difference in the relative incidence of MRSA infections as opposed to MSSA infections between the two levels of society. Yes, sir.
Is it known whether that was an export from the developed world to the developing world, or whether over-enthusiastic use of antibiotics in both places led simultaneously to different forms of...
One cannot explain the success of the MRSA epidemic with the use of antimicrobials alone. alongside with the acquisition of genes that confer methicillin resistance is an acquisition of traits that lead to increased competitiveness and virulence of the organism. And if you take this particular view, then I think it will become apparent that there are different solutions to staphylococcal pathogenesis, which is to say there are clones that you find predominantly in North America. The current MRSA epidemic in North America is to greater than 60% caused by a single clone, whereas in the developing world, these are different clones that have arrived at similar virulence and resistant traits due to the acquisition of mobile genetic elements that confer these attributes. And they're shared between North America as well as the developing world. So you're not looking at one accident that leads to increased virulence and drug resistance in the organism. It can do this from multiple different starting points. Okay. Now, other factors that play an enormous role in the transmission of the disease is the fact that needles must be shared because there is insufficient supply, and the fact that medicines must be shared. So once ampules are open, they are closed with tape and things like that, and they must last for a longer period of time, must be shared with many different patients. That, of course, leads to an increased incidence of staphylococcal infection in that environment. And it is estimated to be the fastest growing infectious disease for the developing world. Okay, now I wasn't sure who I was going to address, and some of you are more concerned with ethical issues than with biomedical research. So I thought it might be wise to put in a couple of slides to define terms such as immunity and vaccine, as well as protective antigen. And for those people that, of course, know that, I apologize. So why don't we just go through this? I took here an image of Dr. Jenner, who was, I guess, the first to systematically apply smallpox vaccination in his practice to illustrate immunity and vaccines. So some naturally occurring infectious diseases, for example, smallpox, endow those who survive them with immunity to subsequent infection. That is, with the same agent only. And ideal vaccines induce the same state of immunity, obviously without causing the disease. Some vaccines have been so successful as to even eradicate human infectious disease against which they protect. And once again, smallpox is a beautiful example for that success story. Now, Staphylococcus aureus, I should say that there are others, polio, diphtheria, tetanus. Staphylococcus aureus infection does not generate immunity. However, staphylococcal mutants may do so. So it's very important for you to appreciate this talk, that you realize that a person is infected with MRSA or Staphylococcus aureus and that survives. That is in that 50% category that makes it through that year, that those individuals will have not walked away with immunity against that particular infection. Okay? The second is the discovery of protective antigens, and of course that must be linked to the counterfeit of Emil von Behring, who was the first to discover that. So what is a protective antigen then? It's thought to be a genetic and biochemical entity within microbes, here Staph aureus, that is both required for disease pathogenesis and, when offered to the immune system of humans or experimental animals in isolated form, is able to generate protective immunity against this disease. Now, the crux of the matter is that the protective antigen of Staph aureus are currently not known. Okay, supposedly, I will reveal them here today. So this is the secrets of the universe revealed. I mean, it's very ambitious for a talk that is also peppered with turkey sandwiches, but I thought I might as well compete with the nutrition. Unfortunately, everything is more complicated with Staphylococcus aureus. So what I'm trying to tell you is, unfortunately, it takes more than a single antigen to do that. Or if there's one antigen, well, then you have to stay awake through the entire presentation to figure out which one it actually is that will do the trick.
When you say protective, who are you protect? Who's the object of that?
Well, I think the text said humans or animals. And it doesn't matter who the...
It's also protecting the bacteria, too, right? Because that's how it does it thoroughly. Well,
by and large, a protective antigen that elicits an immune response that confers protection against the disease leads to the elimination of the pathogen. So if you look through the history of pathogens that have been subject to that, they have been eliminated. Corynebacterium diphtheria is a prime example.
But that antigen is the same antigen that also confers on its pathogenicity?
Yes. The text said it must be essential for the disease process. And if that isn't so, then the laws of evolution will have it that the organism will get around your strategy. Right? It's important to capture that. Okay, so there's a history of staphylococcal vaccines, and by and large, the historic approach to the vaccines is one where companies have a particular scientific expertise, shall we say. So the company knows how to make carbohydrates of bacteria, it knows how to isolate particular proteins, and then what the company does is they simply branch out to different diseases and say, well, I want to do Me Too kind of experiments. And so one of those Me Too experiments was Nabi Pharmaceuticals, recently sold to GlaxoSmithKline, who will try capsule polysaccharides types 5 and 8, which capture 80% of all staphylococcal strains, in a conjugate vaccine, hoping to elicit opsonophagocytic clearance. This vaccine, in fact, was tried already 5 years ago for a phase 2-3 trial that then failed and didn't show any protection against disease. Why companies keep on trying to do the same thing is a matter that I don't want to go deeply into, but since I'm not a company, the University of Chicago have the luxury of taking the high road, right, John? Okay, so while some of these trials are still underway, the biggest one currently is that of Merck Incorporated, which is trying a single surface protein called ISDB. It's a hemophore that removes heme from hemoglobin and allows the organism to grow in infected tissues. The trial is very large. I don't think the University of Chicago is included, but about 20,000 patients in the United States are enrolled in it in an effort to seek to protect patients with elective surgery. And we will, I guess, know in a couple of years whether or not there's any success to this particular strategy. Okay, now I'll structure my talk to first tell you how Staphylococcus aureus infections work. And unless you understand how they work, there's really no point making a vaccine against this organism. Okay. If you inoculate the organism into the bloodstream of people accidentally, and that happens every day in this hospital, or if you put this into animals, then the following will happen. The organism will persist in the blood for a few hours and then clear through the vasculature through all peripheral organs. You can kill animals that you have intravenously challenged and then examine them whether any organ system is exempt from that rule, and you find that none is the answer. Now, if you take any one organism, and we typically take the kidney, then you can ask whether the organism then arrives in this tissue and whether it persists. And the answer to that query is it does, and it persists for as long as you observe it. All infected animals, and that includes people, are unable to get rid of Staphylococcus aureus. And unless there is a medical intervention, they will die from that disease. Okay? Now, in mice that receive no therapy otherwise, that time period is fairly long. It's about 30 to 50 days. And people it's shorter, they, as far as immunity is concerned, are not quite as prepped up as the mice that we work with. So I'll promise to make the lights better. Turn it back on in a second. Anyway, in this histopathology slide, what you're supposed to see is how long it takes after the initial inoculation before the organism sees a lesion that is detectable by pathological anatomical tools, say hematoxin and eosin-stained tissues. And that is about 48 hours. But in 48 hours, you see a lesion that then grows in size and grows, and then in the end is this kahuna here that sits on the capsule surface. So not only is the lesion produced, it also migrates to the surface of the organs. And this is a blow-up of these pictures, and all I want you to appreciate is that on day 2, this lesion is nothing but an infiltrate of granulocytes or neutrophils. And in this infiltrate, there are a few staphylococci. You can only appreciate this if you have John's position here. Now, by day 5, the structure transforms itself. Now the organism will replicate at the center of these abscess lesions, and then the abscess lesions themselves constitute a ring bark-like structure with 4 layers. The first one of which is what we call a eosinophilic pseudocapsule that is this reddish material that's in the black box. Then comes a layer of dead neutrophils, live neutrophils, and again dead neutrophils. It's still impossible to explain why that layering occurs. Okay, now I think this you can see with regular eyes and light. So if you now take a transmission electron microscopic view of these lesions, then you find that at the center is a pure population of staphylococci. There are no immune cells whatsoever. And these are broken tissues, and you can appreciate the pseudocapsule as a layer of amorphous material that encloses the organism in these lesions. And then surrounding this, it's not shown on this slide, are these layers of dead cells. So obviously what the organism accomplished in a matter of a few days is to build a structure wherein it replicates at the center, shielded from the immune system of the host, so that the host's immune cells can actually not reach the organism. Now, the disease progresses until this particular pathological anatomical lesion arrives at the surface of the organ, and then it ruptures. And as it ruptures, the organism re-enters circulation and then disseminates to new tissues and basically repeats the process until the patient or the animals die from it. So, in other words, once the organism sets a hold in infected tissues, there's nothing you can do from a patient's perspective without medical interference. It's a journey that will travel to its bitter end. Yeah?
So the 50% mortality is only because of medical interference?
No, it's with medical interference for MRSA infection. That is intense vancomycin therapy. The patients that don't receive therapy will die no matter what. Yeah. So, and the cost of a patient and how much is put into saving that patient is considerable. But in spite of that effort and drug sensitivity, it's only 50% survival. Okay, now a few years ago we explored this, and you don't need to appreciate anything about this beyond that gram-positive organisms immobilize envelope proteins by a particular biochemical pathway that requires a signature sequence in the protein. And the enzyme that catalyzes is sortase. Anyway, this signature sequence is remarkable for the purpose of this presentation only due to the fact that it allows you to search the genomes of staphylococci for proteins that are deposited in the envelope. It's a game that only takes 3 minutes and you know them all. Okay? So endowed with that knowledge, you can then ask whether particular proteins in the envelope play a role in the disease pathogenesis. You can also, because of this pathway, address the question whether the proteins are important by simply making a mutation in this gene for the enzyme that immobilizes all of these proteins, and then you generate basically organisms that are naked as far as proteins are concerned. Imagine a virus without any protein in the envelope. That is what a staphylococcal sortase mutant is. Okay? And so if you introduce this mutation into the staphylococci, the short end of it is there are no abscess lesions whatsoever, and the organism, while it does get into the tissue, is cleared within a few days, and there's no disease process whatsoever. So while I'm not saying that sortase is a protective antigen, it is, these surface proteins that are absolutely essential for the disease process. And that simplifies matters for what we want to do considerably because it narrows it down to 23 proteins. Okay. So we have the advantage of Dominique Misiakas, who is a professor at the University of Chicago and generated the Phoenix library, which has a mutation in every single staphylococcal gene. And all we got to do then is, once you do the 3-minute search for bioinformatics, you pick out the mutants and you ask which one of these mutants are required for the disease process, and that was shown here. So if you now click on the, this is supposed to be a red box. I think these are supposed to be red. It looks black to me. So whatever is blocked is a mutation in a gene of 23 surface proteins that is required for the disease process. The data for these dots is for day 5 bacterial forming units in the kidneys. So you see that not only do they not form abscesses, but they're also reduced in the bacterial load. So the two actually go together. Okay? Now, a few years back, a graduate student in my laboratory did this experiment. And I thought this was a very important experiment. She said, well, obviously, if these genes are required for the disease process, you could then ask whether they act as protective antigens. And for those of you that are skilled in the art, you know already it's very simple. You purify each of these proteins, inject them into the animal, generate an immune response, and you ask whether you get protection. And this is what this experiment is. And if you look at this, then you see there is no single antigen that leads to a complete clearance of the organism. That, of course, was depressing. Because basically what it meant is, well, the entire cast of the surface proteins is required for this. I can't get protective immunity out of this by taking any one of them. There must be a secret that we don't yet understand that needs to be unlocked in order to capture a vaccine for staphylococci. So, and that is the secret I want to tell you about. All right. Before I do this, I just want to summarize what I just told you. So staphylococci, and it doesn't matter what infection you're looking at, whether it's a bacteremia, it's a skin infection, they all basically do the same thing. There is circulation that the organism exploits to disseminate. It seeds a lesion with neutrophils, transforms this into an abscess that then ruptures and closes the circle. Okay? Okay. Now, this illustrates that there is no protective immunity. If you go ahead, infect animals, and then clear them with antimicrobial therapy, we use chloramphenicol for that, and then come back to challenge animals that have been infected but received therapy and compare the outcome of disease with animals that are completely naive, then there's no difference in the disease outcome. So not only do animals suffer these persistent infections, immunologically speaking, they learn nothing about disease protection from this process, which makes this organism the world champion of immune suppression. It's really remarkable how well it works. Well, okay. All right, so there's a fellow in my laboratory who approached this problem, again with genetics, and he said, well, since the organism survives in blood, what is it that allows the organism actually to survive in blood? And he used molecular genetic approaches for this. So quickly said, whether it's human blood or mouse blood or rat blood, so mouse, rat blood, human, then the organism survives without any decline in CFU. This is amazing. The reason is you could take any other organism and you put them in blood. The blood has considerable killing capacity. They will remove the organism from the blood. But staphylococci, then nothing happens. Now, that includes people that have antibodies to some degree against staphylococci and so on. Now, if you now try individual mutations, then he identified a gene that gave the biggest phenotype. And with this mutation, all of a sudden, staphylococci would get killed within 30 minutes. He saw up to 50% killing of these staphylococci. And so it is this phenotype that he pursued. If you look at a blood smear, then what you observe is that the wild type has the staphylococci actually inside of the immune cells, that is the neutrophils. So this is again fascinating. While neutrophils are the killers of the innate immune system, and they phagocytose staphylococci, they cannot kill them. And the organism stays alive in these cells and then travels with them to the periphery. Okay? Now, the difference in the mutants is that the organisms are, again, intracellular, but now they get killed. And the question is, why would they get killed? Okay, I'm going to skip this in the interest of time. This just shows the virulence defect, and it shows that the mutant doesn't make abscesses, to get to this slide. So the biochemical attribute of this surface protein is that it converts ATP or AMP to adenosine. And why is that meaningful? So this is a thin layer chromatography assay that shows you that when you offer AMP to staphylococcal envelope, then you get conversion to adenosine. And you can do this for staphylococci and for a purified protein. It works equally well. So adenosine is an immune suppressive molecule that we synthesize to end inflammation. Typically, when there's an inflammatory response of whatever kind, the cleanup action at the end of it that terminates the inflammation and allows us to restore the functionality of our tissues requires the production of adenosine. We have an enzyme that is CD71 that actually synthesizes adenosine from adenosine monophosphate, and that is released from cells that get damaged. So there are animals and people that have a defect in adenosine deaminase. They are unable to remove adenosine once it has been synthesized. And that causes the most severe immune suppressive effect known to the medical establishment. So what the organism in essence does is it uses the same immune suppressive molecule that we synthesize ourselves. In fact, the gene that causes this particular thing to happen is called adenosine synthase, is a homologue of CD71. So the bad news about this is that there's actually quite nothing that you can do about the outcome of staphylococcal infections as far as adenosine is concerned, because if you raised antibodies against this protein ADSA, these antibodies might cross-react with CD71. And if they neutralize our own ability to synthesize this molecule, then there might be severe autoimmune consequences to vaccines. Any questions about this? So I cannot but detect a certain beauty in this strategy, because obviously the fellow Staphylococcus aureus figured out how we ourselves manipulate immune suppression whenever we need it. And precisely this strategy was then exploited by the organism. This just shows you that when you inoculate the organism in blood, that it synthesizes extraordinary quantities of adenosine that float around the blood of infected people. So hitherto that hasn't been detected simply because people don't take the blood of bacteremia patients and put them over an HPLC. But if you did this, then you would find that there is massive quantities of adenosine that is synthesized by bacteremic individuals. All right. Now, if you remove ADSA, then you get clearance of the organism intracellularly. So the model is as follows, that the production of adenosine inside of the neutrophil is what protects the organism from the killing, and there's unfortunately nothing that you can do about this. So all this research so far was no good as far as protective antigens was concerned. Okay. Now I'm going to tell you about a molecule called protein A. So it so has it since 1956 that staphylococci are known to develop an antibody, if you wish. And what the organism does is it expresses a protein that has been called protein A, and that binds to immunoglobulins and decorates the organisms with it. This is a crystal structure appreciation of that, and for you this is not very insightful because it's too far away, but it suffices to say that the Fc portion of antibodies are captured by protein A in the envelope of the organism, and in addition, it binds to the Fab portion. Now, what is that? The Fab portion is the portion that is presented in the vicinity of the antigen binding site, and when the organism's protein A cross-links the Fab portions, it acts as a B-cell superantigen for Staphylococcus. So this shows you the mixing of Staphylococcus with immunoglobulins. You see this green signal is nothing but immunoglobulin. This is the structure of protein A. It has 5 immunoglobulin binding domains. All right. So even if there are antibodies that are present, the organism will coat itself with these antibodies and thereby evade protective immunity. So we took a structural biology approach where we said, why don't we make a molecule, protein A, that can no longer bind to immunoglobulins, but otherwise has the amino acid sequence preserved. Okay, the rationale is the following. A gene is required for disease establishment, and if you made antibodies that neutralize protein A, the ability to sequester all of these immunoglobulins would be neutralized. Okay, and that turned out to be the key to what I believe is a success for staphylococcal vaccine. Now, we started out with a single immunoglobulin binding domain, introduced 4 substitutions in it, and they would, on the basis of this design, abrogate the ability to associate with IgG. So this is wild-type protein A, and this is the wild-type SPA-D domain, and this is a mutant SPA-D domain, and then we put IgG over it. You can see that wild-type and the SPA-D domain capture IgG, but the mutant does not. Okay, so it works like a charm. And this is a quantification of the same that shows that the mutant can no longer bind to immunoglobulins. Okay. Now, this is an assay for the B-cell superantigen activity. If you inject protein A into an animal, what happens is that the B cells in the spleen begin to die within a few hours. And so the rationale for that is that the protein cross-links the B-cell receptor triggers the immune cells to replicate, and then, because they lack the appropriate stimulatory signal, they commit apoptotic collapse. The beauty of this strategy for the Staphylococcus is the following. When you get infected with the Staphylococcus aureus, you recognize the antigens, but the organism kills off your B cell response, so in effect, you don't make specific antibodies against the organism. Okay? If this were dark, then you could see the apoptotic B cells here, and you can't see this for this mutant protein. Now, does this really work? And so what Han Kim, a graduate student in the lab, does, he purified antibodies against protein A that are raised against this non-toxigenic variant, and he cut off the FC portion to purify the fat portion. And that fat portion is purified here. And he asked whether this fat portion, which now can only bind to protein A as an antigen, but not to these other attributes, can block the association with protein A with the IgG or von Willebrand factor, another ligand for protein A. And I think you can appreciate that it does inhibit it. So lo and behold, the antibodies that you make in this process are actually specific for protein A. They bind protein A as an antigen. Okay. So we also made a full-length version because this was a much more powerful antigen. And now I want to illustrate what staphylococci do to you. If you sample humans as they would sit in this room and you ask whether or not you made antibodies against protein A, and I guarantee that all of you have had a staphylococcal infection, then the answer to the query is that there are no antibodies against protein A in your blood, in spite of the fact that this is the most abundant protein in the envelope of the organism. Now, for those of you that received the diphtheria vaccine when you were a child, you still have massive amounts of antibodies 30, 40 years after immunization. It gives you an idea what memory responses you could get for some antigens. Okay? So it's not that these people cannot make antibodies. Now, if you infect a mouse with staphylococcal strains and you ask whether as a result of the infection they make antibodies against protein A, then the answer to that query is also no. But if you now immunize them with this protein A that is a non-toxigenic protein A, then they make massive amounts of antibodies. Okay? So what do these antibodies do for you? The interesting aspect is this molecule here. This is a full-length non-toxigenic protein A, and we're testing this against the MRSA strain that's a current epidemic clone in the United States. And the CFU, 5 days after infection, dropped by 3 orders of magnitude, and the abscess formation is eliminated as a consequence of that immunization. Okay? This is the best part, and there's something wrong with the color. You can't see it. Anyway, what is supposed to be red turns out to be black on this projector. And the other bars are blue, but I'm not sure you can distinguish blue and black. It doesn't matter. It doesn't matter. If you just follow this, every one of these letters represents a staphylococcal antigen that was purified in recombinant form. And when we infect an animal, what we ask is whether the animals make antibodies against any other antigen other than protein A. And it turns out that they make antibodies against one protein only. It's this red-black bar. It's called EAP. If you compare all the other signals, there's either nothing here, or there's a tiny little bit of antibody for 4 antigens. So the organism is programmed to cause an infection where the humoral immune response is limited to a single antigen. If you now immunize these animals and infect them with the same strain, and the immunization neutralizes protein A, you make all these other antibodies. So what the vaccine actually endows you to do, it endows you to elicit an immune response against the organism, and that immune response is protective. It's not just protective because of protein A, but it is protective because it enables the vaccinee to make antibodies against staphylococci. I believe that that may be the key to making a staphylococcal vaccine. Okay. How am I doing with time?
Another 10, 15 minutes.
10, 15 minutes. Okay. So I think what I showed you in the last 10 slides or so are 2 examples for the immune evasive strategy of the organism. That is, it has no interest to be killed by immune cells, and it has no interest in the infected individuals to mount a humoral immune response. And I suspect that you can get around this by simply vaccinating against protein A and neutralizing this ability. That is not to say that protein A constitutes protective immunity. The question rather is what does the organism have to do in order to establish the disease? The immune suppressive attributes that surround this are just there to ensure you can do this forever. But it's not actually the mechanism that establishes the disease. So the mechanism that establishes the disease, I think, is displayed on the next 5 to 6 slides. It is known since 1908 that all staphylococci produce a coagulase. And when Karen goes ahead and she isolates a staphylococcus in the bloodstream and says, well, it's a gram-positive cocci cluster, the next thing that you want to know is whether or not they make coagulase. Because then you classify staphylococci as coagulase positive, i.e. Staph aureus, and coagulase negative. These are opportunistic pathogens that don't cause disease in healthy people. All right? So from that very fact, you actually know already since 1908 that there's a distinction in virulence between staphylococci that elaborate the coagulase and the other guys that don't do that. If you look in abscess tissues and you use antibodies against host proteins now, not bacterial products, then you find that they coat themselves with a eosinophilic pseudocapsule that is made up of 2 molecules, prothrombin and fibrin. Okay, so these are clotting products of blood. And that is, in fact, what the organism does when you inoculate it into plasma and you inhibit thrombin even. This organism will clot the blood by secreting 2 proteins, coagulase and a second protein called von Willebrand factor, binding protein. Okay, this shows you a diagram of the 2 molecules, and they are beautiful to behold. The first one of which, the coagulase, has a domain D1, D2 that binds to prothrombin and activates coagulation by generating a new active site that operates this molecule indiscriminately of the concentration of calcium. Calcium, as you know, is the physiological regulator of thrombin activity. Now, at the C-terminal end of fibrinogen binding sites, they bring the fibrinogen to this molecule that is complex with prothrombin, and then this molecule will convert fibrinogen to fibrin and generates a clot. The product of this clot is different from the physiological clotting product that occurs with thrombin and is believed not to be degraded by TPA and other factors that would allow you to dissolve otherwise the clot. So the organism generates its own clot and renders the host unable to dissolve it. The second molecule is called von Willebrand factor binding protein. It has a similar D1, D2 domain and catalyzes the same reaction. It captures fibrinogen by a different module. If you mutate the 2 genes, and that's evidence for that is here, but what happens is, I think you can see it here, the blood no longer clots when you infect it with staphylococci. Wild-type staphylococci will clot the blood, and that's it. That's all there is to it. But without these 2 genes, there's no clotting whatsoever. Okay, what's the virulence defect? The virulence defect of a coagulase double mutant with von Willebrand factor binding protein is that there are no abscesses whatsoever. Presumably because the eosinophilic pseudocapsule cannot be established. In a lethal challenge model, the animals have the ability to survive for a very long period of time. And if you just measure the survival in blood, there's even reduced survival in blood without the coagulases. So I surmise that the coagulase is actually the key to establishing the lesion that I showed you, namely these abscess formation. All right. Now, there are 2 ways how you can do vaccines. You can either purify the antibodies, passively transfer them, and you can also do this by active immunization. So if you do this by passive transfer, you can, through antibodies directed against both von Willebrand factor binding protein and CoA, protect against abscess formation. And the same is true if you do it by active immunization. You eliminate all abscess formation. Okay. So what I believe, at least today I do, is that protective immunity against staphylococci is the correlate for which is the ability to block coagulation. The reason why that doesn't occur in a physiological infection is that the organism prevents the generation of antibodies against coagulants. Okay? I'm going to skip all of this and tell you this, unless we do for questions. So obviously, this is all benchtop research, and the question is, how useful is all this? So I made a few bullet points to get at what I just said here today. Antibodies against multiple antigens are required to generate protective immunity against Staph aureus infections. I didn't talk about this bullet point, so we can ignore it. The coagulases are protective antigens for bacteremia and soft tissue infection of Staphylococcus aureus. Now, the sortase-anchored surface proteins and coagulases are essential for the pathogenesis of the disease process, and protein A vaccine prevents staphylococcal ablation of B-cell responses and provides for disease protection. The simplest staphylococcal vaccine is probably to immunize newborns, or at least infants, with the non-toxigenic protein A. You probably don't have to do anything else, because on every subsequent exposure to the pathogen, the vaccinees should make antibodies against staphylococcal proteins. The initial disease might still be associated with some morbidity or mortality, but eventually people that survive the initial exposure will make antibodies, and that should reduce the disease burden against staphylococci tremendously. Now, if you want to be picture perfect and you want to ensure that every vaccinee is protected against every disease, including the first one, I suspect you need more than protein A, because the protein A vaccine is predicated on a functional immune system and the exposure of the pathogen. So in those scenarios, you're going to have to throw in the coagulase and von Willebrand factor binding protein so that the vaccinees have already the initial correlate of protective immunity. Okay? Now, the people that did this work, have you been to this site? This is in Crete. This is a wall, the Law Codex of Garde, that is 3,000 years BC. So they put this on the wall. I love that site. And anyway, I took a picture. I always use it. And I put the names of the people that did the work on top of that, hoping that their names will live in the same perpetuity as the law code of the Greeks. Anyway, so Alice Chang, Andrea Dedent, Han Kim, Molly McAdow, and Viltama Vongsa are in my laboratory currently and work busily on these things. And they have done all this work. Yukiko Stranger-Jones and Julie Bubeck-Wardenberg used to be in my laboratory, and they have done good things, but they moved on. Thanks for your attention.
Jesse.
I think as you presented it, if I understand it, there are no immune differences that you highlighted between MSSA and MRSA.
No.
While there's a big difference in our ability to treat them effectively with antibiotics, are there some additional routes that MRSA has or employs to be a more aggressive or invasive infection that are relevant here?
So I didn't get a chance to talk about this, but obviously there's 20% of the genome that is different between these strains. And that difference is made up by either pathogenicity islands or bacteriophages that have lysogenized the organism. They do carry virulence genes, and I think the sum of these elements gives rise to differences in the relative virulence attributes of Staphylococcus. Now, the trait to express coagulases and protein A is not a variable trait. They all do this. There isn't a single staphylococcal strain known to have a protein A null mutation that has been isolated from a clinical case. And as far as coagulases are concerned, they all do express coagulases to variable degrees, have two genes for this. And there might be strains that are negative for one of the two coagulases, but they are very rare. I mean, these traits are so common that we use them for diagnostic purposes of isolated variants.
To the extent there is this variable repertoire of the subtypes, will that cause the vaccine to be an evolutionary pressure to favor them?
So,
just like
there are two
antibiotics.
answers to this, Jesse. The first answer is that as far as protein A is concerned, the immunoglobulin binding domains are non-variable. There is a segment of protein A that has a variable segment to it, but that is not part of this B-cell superantigen attribute. It's very interesting. There's a suggestion that this peptide leads to the uptake of a segment of protein A. This is something that Alice Prince works on in Columbia. I didn't have time to get into that. But I suspect that there is no variation in protein A, and there doesn't have to be because the molecule just works the way it works. There is variation in the coagulases, and one has to address that.
Dan, you have a question?
No, I was going to ask about if the protein A doesn't meet the criteria to be a protective antigen, then would there be evolutionary pressure to create a
Well, so the way I look at pathogens, you could, broadly speaking, categorize them into two kinds. Those that are immune suppressive, that elicit no protective immunity or immunity following infection, and those that do. The latter group engage in variable sets of protective antigens. Group A streptococcus, for example, the key protective antigen is M protein, and that is the gene that is varied, and that for that reason there has never been a group A vaccine because there's 100 different types. But Staphylococcus aureus is not in that category.
Yeah. I understand that MRSA is considerably less in Germany as compared to France, but both of them are scientifically developed equally. And they say the behavior of the doctor and expectation of the patient makes a big difference. Mm-hmm. Germany has multiple times more, less than France has, and I just wondered if
I have a view on the matter.
Awareness.
Well, I'm German. So. Well, I think the reason for German and Dutch success in keeping the MRSA epidemic in the hospital at bay has something to do with the discipline of the physicians. That's they're a funny bunch, but it works out for some things, right? Now, can you duplicate this in France? No, it's just more fun to be in France. So.
So, are there host factors or even environmental factors that may determine how people react to, when you try to vaccinate? Because as you move from, developed to developing countries, are there host factors?
Well,
parasites, things like that.
I suspect that there are host factors.
Yeah.
So Mycobacterium tuberculosis is another organism that doesn't leave protective immunity on infection. The immune suppressive strategy is thought to be a type 7 secretion system, although it's not truly known whether that accounts for all of it. And the mechanism for protective immunity also hasn't been revealed. In that particular scenario, it's very well known, though, because the disease lasts so long. And you have the possibility of measuring infection through the skin test, that there are host factors. There's roughly a third of the people of this world that are resistant to MTB. And there's a third of the people of this world that are highly susceptible, and then another third that get infected but can mount some form of immunity so that they live with the organism for some time. The reason why I give you the MTB answer is if similar traits existed for diagnosing staphylococcal exposure and infection, one could address that. Currently, you cannot address this. There's some of us that get small staphylococcal infections, but these people may actually be immune to the disease, but we just wouldn't know it.
Please.
Is there a role played by quorum sensing?
Yeah, there is a quorum sensor, and the organism elaborates a peptide that it constitutively secretes. And that is then perceived by a two-component regulatory system. The system is called AGR. It so turns out that when you knock out this quorum sensor, that bacteremia and abscess formation are still functional. And there's been a gentleman at NYU who proposed that one could use the quorum sensing system for a vaccine approach. But I haven't seen any evidence that that is viable.
How close are we to clinical trials?
No, I collaborate with Novartis Vaccines, and the question is, where, when, how to put this thing into. It has to go through safety trials, The protein A you probably don't know about is a toxin. So it's been accidentally introduced into people as an RSV fusion protein. And that actually led to considerable side effects, immune side effects in vaccinees. And obviously FDA would not allow to use wild-type protein A to be introduced into people. So the hurdle before us is to convince FDA that this non-toxigenic protein A is indeed non-toxigenic.
What are your thoughts about the environmental impact has been very different in the third world and the supposedly developed world, and possibilities for addressing that.
Well, I think what happens, Mary, when you immunize with any vaccine is you might, with some luck, eradicate that vaccine. The problem with that approach is that another organism will take the ecological niche of that organism. So a prime example is Haemophilus as an oropharynx colonizer and disease pathogen. The eradication of that, the substitution with pneumococci, now the eradication of pneumococcal types, and the substitution with Staphylococcus presumably. So I think it's a game that we play between microbes and vaccinees in that we change the colonizing spectrum of people that have been vaccinated.
Yeah,
Is it a difference?
From Novartis.
What's that?
Getting your own grant support from drug companies.
Yes. It's an ethical problem.
To what extent, if any, does the incidence in the developed world as opposed to the incidence and causes and variation in the developing world have an impact on your application for support?
So, I'm actually the wrong person to talk. There's a talk tonight at 5 o'clock where Rino Rapopoli, the global head for vaccines and diagnostics at Novartis, gives the Andros lecture. I urge you all to attend. It's a fascinating talk and a fascinating scientist. What Rino has done is he's the head of Novartis Vaccines Diagnostics, which is the commercial branch, and he has founded the Vaccine Institute of Novartis, which is a non-for-profit organization. The problem with vaccines for the developing world that stand no chance of making any profit is that people that don't have the means to make vaccines may have a good spirit, but they're not able to make vaccines. That's just the way it is. So, the idea is to spin off the abilities of Novartis for this nonprofit organization and bring vaccines actually to the developing world. He has something to say about this. I don't know enough about this particular matter to comment on it. What I want is a protein vaccine that is as stable as a rock, survives in tropical countries for a year in a glass, and you just need to take a little bit of sipping water, put it in there, mix it up, and inject it. And if I can do all that for 3 cents, that would be the way to go.
Mary's question is a nice segue into, just before thanking Professor Schneewind, announcing that next year's seminar series, still to be finalized, but the topic is settled, is disparities, local, national, and global. And the possibility, indeed, of coming back to some of these questions of disparities on the global scene is one of our plans. We will resume this series the last Wednesday in September, which I believe is September 29th, which is the first Wednesday of the fall quarter. Please join me in thanking Professor Schneewind and the